We will get started. So first, let me introduce myself. I'm John Morris, and I have the honor and the privilege to direct the Knight Alzheimer's Disease Research Center and its Memory and Aging Project. And we are, all of us at the Memory and Aging Project are just really so thankful for all of you and for coming, for all you do for us in terms of our research and your participation, but also coming today so we can give you some information about the research that we have been conducting. You can find out some of the results and also tell you about some research that we're going to propose for the future. Now you have at your seat several things, including a copy of all of the slides that will be shown and including the program. Also at your seat are name tags and should be index cards, and you should feel free during the presentations, if you have a question, to write it down on the card. And at 10:30, when the presentations end, we'll collect the cards and the questions. We'll have all the speakers up here, and we'll try to get through as many questions as we can in the time that we have available. So first points are outside those doors, and I think to the left are the restrooms. So make sure that we have Memory and Aging Project personnel around. They can direct you if you have questions. Second, if you have a cell phone, will you please put it on vibrate or turn it off? And third, feel free at any time to get up and help yourself to coffee or whatever you like. Oh, good. We got there already. So this is the faculty and staff of the Knight Alzheimer's Disease Research Center. We have at Washington University School of Medicine 45 different faculty who participate with us. We have 85 staff, and we're very interdisciplinary. It's not just neurology or psychiatry or radiology or geriatrics, but we have people from many different disciplines participating in the Alzheimer's Disease Research Center, including somebody from obstetrics and gynecology. Now, I'm not quite sure how we got that person in, but and I'm very biased, but our faculty and staff are the best in the world. And I'd like all our faculty now to stand up, please. So this means Jason, and Jeff, and Sarah, and Virginia. They are terrific. And I know that our staff are permeated throughout the room and outside, but all the Memory and Aging Project and ADRC staff, if you'll come forward and just raise your hand so people can acknowledge you. So staff, stand up, raise your hand. I don't know if you notice, they really don't do what I say. But in real life, we all work together. We're all very happy. Now, putting together a participants meeting such as this is no small task. Everyone that you see here have been contributing to this, but I want to point out a few people who took the major responsibility for this, and that's individuals in our outreach, recruitment and education corps. That's Andre Denny, Jennifer Phillips, and Murtis Spencer. So let's give them a round of applause. Also, for our audiovisual work of Ron Hawley and Howard Palmer, they're seated over there at the technology table. And finally, Krista Mulder and Maria Carroll for their leadership. So thanks very much. All told, we have about 900 research volunteers that we see annually in the Memory and Aging Project. About half of you are here today, so that's terrific. We have turnover from time to time, so you see some individuals that are saying goodbye or have said goodbye to go on to another position. I'm going to talk about two of them in just a moment, but we have some new individuals as well, as shown here. And I know some of them are here. I know that Stacy, Marissa, Megan, and David for sure are here. If they're here, will you stand up, please, so we can... There's Marissa and Stacy. Where are you? Stacy's here, and Megan and David. David's in the back waving his hand. Megan, so let's welcome them. So these two individuals are leaving. They're retiring. So Gene Rubin is a psychiatrist. He and I started way back in the 1980s in the Memory and Aging Project, doing clinical assessments and analyzing data and writing manuscripts. Gene and his wife Dottie and their family, and Lucy, my wife, and our children have been friends for many years. And Gene is starting his phased retirement July 1. So I joined the ADRC in 1983. And Gene joined it in 1985, and Betsy Grant and Mary Coates in 1986. So Gene is stepping down, and so we'll miss him very much. And then Virginia Buckles is seated here next to Joanne Knight, and she's been an administrator coming in 1992 to join me and really help me in my career. Couldn't have accomplished nearly anything without Virginia's help, and she rose to be the executive director of the Knight Alzheimer's Disease Research Center. She relinquished that role about a year ago, and Krista Mulder now is the executive director, and Virginia is going to take retirement at the end of this month. So let's thank Virginia for all of her efforts. So here are today's presenters. We're going to hear from Lenice Cummings Vaughn about predicting cognitive decline, predicting dementia. Who's better at that? The person who we are enrolling and assessing in the Memory and Aging Project, or their study partner, the person who comes with them and we ask questions. So Lenice is going to tell us about that. Justin Long is going to talk about a hot topic: can infections produce Alzheimer's disease or cause dementia? So he's going to review that. Then David Holtzman, who is the chair of the Department of Neurology, is going to talk about developing therapies, because his laboratory has been at the forefront of developing potential therapies to treat Alzheimer's disease. Dave had a travel issue last night. He couldn't make it into St. Louis as he originally planned, but he is scheduled to arrive at 8:50, and hopefully will make it here. But if not, he was kind enough to send his wife Tracy, and she can present it. And then Susan Stark, Virginia Buckles, Mitch Summers, and Jeff Zox are going to talk about some newer studies that we're contemplating, so to give you a heads up. Outside in the little lobby area, as we have done for the past several years, there will be some personnel and some brochures talking about ongoing studies that we are doing. So we won't be presenting these here today, but there will be, after the meeting closes at 11, there will be people outside to talk about various studies that you can interact with the staff and get more information about and ask your questions. I want to come back to this. You notice that Dave Holtzman is scheduled to present on the therapy of Alzheimer's disease. Normally we have Joy Snyder. present on the therapy of Alzheimer's. Joy is seated right here. I want to tell all the speakers, and we've done this, we've gone over sort of the ground rules of speaking, and keep everything on time and so forth and so on. But I didn't mention one ground rule, so I better bring it up now, because last year, Joy Snyder went rogue on us. So here's the ground rule. I tell the jokes. Last year, Joy tried to tell some, and you notice this year she's in the audience, not a speaker. So here's a conceptual framework for what we think about Alzheimer's disease. So when people say Alzheimer's disease, almost universally we think of somebody who has developed the memory and thinking problems, the dementia that Alzheimer's disease causes. That's what we think about. And we call it here symptomatic Alzheimer's disease. It begins very mildly, but always gets worse and always ends in death. That symptomatic phase of Alzheimer's lasts about 7 to 10 years. But what we've learned is the longer period of Alzheimer's disease begins in the brain at least 20 years before any symptoms are produced. As a matter of fact, it begins with changes in some proteins. Some of you have heard of amyloid beta, and we can measure that in the spinal fluid and by PET imaging, also tau proteins. And what this slide indicates is as these changes build up, they haven't caused enough brain damage to produce any symptoms. These individuals are perfectly normal in terms of memory and thinking, but eventually they build up to the point where the brain cells, we call them neurons, no longer can tolerate this and they begin to die. And this is denoted like this. This is the normal number of neurons, brain cells we should have, but eventually they die, and when they die, the symptoms occur. Does that make sense? Okay? We've been trying to treat people with the symptoms, and we will continue to try to treat people with the symptoms. But guess what? Brain cells already are dead. We're dealing with a brain that has about half of the brain cells that it should. So we're still working on therapies for people with Alzheimer's dementia, but we've also started studies here, trying to treat with drugs in this phase of the illness before there's significant brain death, cell death, and maybe we can delay or even prevent Alzheimer's. Does that make sense? So that's what all of our studies are really organized around. That's why we ask for the imaging studies, for the spinal fluid studies, for the blood studies, because it's telling us those brain changes that we can track, hopefully, to prevent Alzheimer's disease. When we look at the spinal fluid and we look at the images, we're looking for biomarkers, and we think they identify Alzheimer's disease before symptoms. We call it preclinical. We're asking a number of questions. If you have a positive biomarker and you're still mentally intact, does it mean if you continue to live, you automatically are going to develop Alzheimer's? Or are some people protected against that? Alzheimer's dementia, we don't know that. So we're following people. When does the person change from being perfectly normal to developing the symptoms of Alzheimer's disease? And can we use trials of experimental drugs, investigational drugs, in this preclinical stage to delay or prevent Alzheimer's dementia? Now, our work is supported largely by grants from the National Institutes of Health, primarily the National Institute on Aging. And one of those grants is called the Alzheimer's Disease Research Center Grant. But our program began in 1979 under the direction of my predecessor and mentor, Leonard Berg. He started a study in August of 1979 to do clinical evaluations of healthy older adults and older adults with mild dementia caused by Alzheimer's disease. He called that the Memory and Aging Project. And that's where all of you come for your assessments, the Memory and Aging Project. The Memory and Aging Project isn't a specific grant, but it's supported by the grants that we do, and it's our clinical research office. So in addition to this Alzheimer's Disease Research Center grant, we have two other grants from the NIA, one called Healthy Aging and Senile Dementia. Now, I'm looking around, I don't think anyone here is old enough to remember that back in 1984, a term for Alzheimer's dementia was senility. And that's where this comes from. This started in 1984. They didn't even know it was Alzheimer's disease. And then the adult children study. These grants are the primary support for all the studies that we do. But we have other terrific support from individuals who want to make a difference and do so at a tremendous support mechanism for the Alzheimer's Disease Research Center. And in 2010, Charles and Joanne Knight endowed the center with a very major gift. So we call it the Knight Alzheimer's Disease Research Center. And Joanne is with us today. So let's thank Joanne. We added another grant called the Dominantly Inherited Alzheimer Network. This is a study of the rarest form of Alzheimer's disease. This form of Alzheimer's disease is caused by a single bad gene that people inherit from their parent. If they inherit that single bad gene, we call it a mutation, that offspring will develop Alzheimer's disease 100%. And usually at a young age. On average, 45. But we have people in their 30s and their 20s who have developed Alzheimer's dementia because they inherited this rare gene that causes the illness. Now, this form of Alzheimer's is less than 1% of all Alzheimer's disease, so it's very, very uncommon, which is why we have to go around the world to get participants. And I'll just show you that the DIAN, Dominantly Inherited Alzheimer Network, has sites in North America, Europe, Australia, Korea, and Japan, shown here in red, and new sites being developed in Central America, Mexico, the Caribbean, Puerto Rico, and South America. We have a site already in Buenos Aires, and we're developing other sites. So we need to have this global reach to recruit and evaluate appropriate individuals. And this is a very important study to understand how the disease produces, the bad gene produces the brain disease that produces the dementia. We have many partners in the Knight Alzheimer's Disease Research Center. The St. Louis chapter, it's no longer the St. Louis chapter, Greater Missouri chapter of the Alzheimer Association. And we have Adrienne Holden here from the Greater Missouri chapter. Let's welcome her. And two very important advisors to the Knight ADRC, our African American Advisory Board, and members of the Links Incorporated, the St. Louis chapter. The African American Advisory Board has been advising me and others in the Alzheimer's Center since about the year 2000 to make certain that our research is welcoming to people of color. If we only study Alzheimer's disease in white people, we'll only learn about Alzheimer's disease in white people, so we need to be certain that our research volunteers reflect the diversity of the St. Louis area. We have the vice chair of the African American Advisory Board here, C. Jessel Strong, but I'm going to ask not only Reverend Strong, but all members of our African American Advisory Board and members of the Links to please stand up. And let's thank them. We have at Washington University a Center for Aging named for Harvey Friedman, the same person who endowed my professorship. Dave Holtzman has established the Hope Center for Neurologic Disorders. We get great support from Barnes Jewish Hospital Foundation and from Washington University, including from the Chancellor, the Dean of the School of Medicine, and I mentioned our department chair, Dave Holtzman. We have great support from Alumni in Development, and Alex Carr is here. Alex is standing up. You might notice this. And let's thank her for her work. And I've already mentioned we have terrific philanthropy. Joanne and her family, the Knight family, continues to support our work. And just this year, we have a very generous gift from Roger and Paula Riney. And Roger, would you mind standing, please? Let's thank Roger. Well, a number of our faculty, remember, they're the best, so they garner awards. Randy Bateman received from the American Academy of Neurology last month the Potamkin Prize. It's the, quote, the Nobel Prize of Alzheimer Research. Ann Fagan, who leads our biomarker corps, received from the Academy of Sciences St. Louis the Trustees Award. Eric Music and Jason Hassenstadt, both with us, come July 1, have been promoted to Associate Professors of Neurology with tenure, very important. Brian Gordon is a Blumenfeld awardee, and Lenice Cummings, Cummings-Vaughn, who you'll hear from very shortly, received the Coppola Award. Let's congratulate all of them. It turns out Lenice is not only a scientist, she's something of a socialite. There she is in the Ladue News of May earlier this year. Let's talk about you, the Memory and Aging Project volunteers. So how many do we have? Well, it depends on how you count it up, but let's just consider people 65 and older, so 537. The Adult Children Study enrolls middle-aged people beginning at age 45, and that's another almost 300. Ranging in age from 20 to 103. Remember that DIAN study, sometimes those gene mutations cause the illness in the 20s, so we need to enroll people as young as 20. So we're really looking at the illness and the brain health throughout the adult lifespan. 57% of you are women. I didn't actually count this morning, but that's what the statistics show. 16% are African American. The longest period of participation is 32 years, and the second longest is 29. And can you believe it that Ted here today is 32 years? Raise your hand. People come from all over the United States, including from California and from Maine. These are some statistics. Most people are cognitively normal, as designated by the heading CDR0, and on average are age 76. The two columns labeled CDR 0.5 and CDR1 are mildly symptomatic Alzheimer's disease. You can see that on average people are 76, 78 years of age and well educated on average. I've mentioned several times we do imaging to identify these biomarkers even before symptoms occur. These biomarkers require MRI scans. Now, MRI is great for lots of things. It shows the brain's anatomy in exquisite detail, and it doesn't require radiation. The magnetic means there's a magnet, so no radiation is generated. When you put your head in that big donut, it's a magnet that you're putting it in. But that raises other issues, including we can't have someone who has metal in the body go into that scan because the magnet might pull that metal aside. So if you had a cardiac pacemaker, you don't get an MRI, right? And some people find putting their head in that donut hole can be claustrophobic. But look at all the items there that our imaging crew will ask you before you get an MRI scan if you have any of these examples of metal in the body. And this includes tattoos. neurostimulators, anything. So they're going to ask you about that. And also for MRI, some people don't like this, but we have to know if you're taking a transdermal patch with medicine, does that transdermal patch have metal? Or do you have a partial plate, dental plate? And so forth and so on. So I'm just telling you why we ask these questions of you. It's to protect you. We don't want the magnet to do damage to you if you have these metal-containing devices. This is a wonderful time in Alzheimer research. Look here, about 2012, the total amount of NIH dollars given to Alzheimer research was $500 million. You know, Alzheimer's disease, Adrian and the Greater Missouri Chapter of the Alzheimer Association will tell you that Alzheimer's disease each year costs us over $200 billion just to care for people. So devoting $500 million to research was ridiculous. But look what's happened. The legislatures, and including, have to give credit to our senator from Missouri, Roy Blunt, have appropriated a large increase in the amount of research dollars devoted to Alzheimer's disease. So now it's $2.4 billion, and that's terrific. We still need more. We've been very successful at Washington University in getting these research dollars. So if we totaled everything up in fiscal year 2019 for Alzheimer-related funding from the NIH at Washington University, it's about $125 billion. How many is that? Million. But to put that in perspective, look at our sister universities in the area that do Alzheimer research. So as I say, we have been very, very fortunate. And again, it comes back to we have the best faculty and staff. We had all of our grants they're given to us for 5 years of funding, at which point we have to reapply for funding for the next 5-year period. And the approval of our renewal application depends on whether we've made appropriate progress and did what we said we were going to do. So we just had to put in last month a renewal application for the Knight ADRC, and we hope it will be funded, and we expect it will be. It always has been. It will then start for the next 5-year period, next May 1st, 2020, and that will be budget years 37 through 41. The total budget for that grant that we put in for those 5 years is $15 million. And we have a nice short application of only 684 pages if anyone wants to read it. You are very valuable, certainly to us, but because you are so committed to research and you give so much of your time and your effort and your imaging, your blood and your spinal fluid, people throughout the world know about you. And we got 289 requests over these past 5 years to either have you participate in additional studies or for other investigators to look at the data that you've provided or your tissue so that they can do their research. And this way, we were able, when we granted access to these resources, the investigators got an additional $43 million in grants because of you. Almost 600 scientific papers published in the last 5-year study. I've mentioned the generous support that we have, and we're using this this support to enable us to do projects that we couldn't do through the funding from the NIH. One of them is to establish a clinical trials unit to expedite the discovery and the testing of new therapeutic compounds. Dr. Joy Snyder is going to head up that clinical trials unit. Again, people around the world know about you, and when investigators from Melbourne, Australia, Johns Hopkins in Baltimore, University of California in San Francisco, Lexington, Kentucky, University of Southern California, Los Angeles, put in grants. They ask us to have access to the data that you have provided because it is so valuable to them. So you've contributed worldwide to Alzheimer's studies. And we have 2 additional studies that we've been doing recently. Dr. Greg Day, who was introduced earlier, is leading a study of early onset Alzheimer's disease. This is Alzheimer's disease dementia that begins before age 65 and is not caused by one of those mutations that I mentioned. And I don't know if people know that Down syndrome is a cause of Alzheimer's disease. It's been said that every individual who has Down syndrome will develop Alzheimer's disease by age 40. And the reason is, we think, is because in Down syndrome, the individual inherits, you all remember your high school biology, when we are conceived, we get genetic data from dad and genetic data from mom, and we're supposed to get one set of genetic data from dad and one set from mom, so we have pairs of genetic data. But in Down syndrome, something happens. Usually dad still gives his one set of chromosomes with genetic data, but mom somehow gives 2. So the individuals have 3 copies of certain genes where they should have only 2. One of those genes is the gene responsible for producing this protein I mentioned, amyloid. So ever since Down syndrome people are conceived, they're overproducing amyloid by 50% all their life. So they develop Alzheimer's disease. Bo Ansis, who is here, and John Constantino are leading our site's involvement in looking at Alzheimer's disease in aging Down syndrome. Mentioned our desire to be welcoming to all people in the Greater Metropolitan St. Louis, including African Americans. We hosted a national workshop last October where we had over 300 attendees to understand how we can improve African American participation in Alzheimer research. Each Alzheimer's disease center, there are 31 of them throughout the country, sent representatives, and it's followed by a publication that we find that those biomarkers that I was talking about that starts preclinical Alzheimer's disease and builds up over decades before it produces dementia, Alzheimer's dementia, some of those biomarkers differ by race. So if we just assume that everyone had biomarkers that we study in white individuals, it won't apply to African Americans, and by extension, we don't know this, but to Hispanics or Native Americans or Asian Americans. So we need to be inclusive in our studies. I'm presenting this quickly for Jason Hassenstab, who leads our neuropsychology and cognitive core. Many of you will recall for your annual assessment, you come to the Memory and Aging Project, you see a clinician and a nurse, you often give blood, and then you come back in a few weeks to get testing of memory and thinking. We call that a cognitive assessment. And that lasts somewhere between an hour and a half, and sometimes it's difficult to get through, but it's very important for us to know how people are doing in their memory and thinking over time. But Jason and his colleague Andrew Aschenbrenner has developed a new way to test cognitive ability that doesn't require people coming to the Memory and Aging Project. It'll be on your smartphone, an app, and it will ask you, if your smartphone gives you a buzz, that you will be tested for 4 times each day for 1 week. But it's less than 3 minutes, a test. So you use your own personal smartphone. If you don't have one, they can help you with that. But the idea behind this ambulatory research in cognition is you'll be at your home, at work, at the Blues game tonight. you'll be in the natural setting where your memory and thinking is supposed to work, rather than what happens when you come to the Memory and Aging Project, which is artificial, right? So this is natural testing, and it's very brief, test often and everywhere, short, and we'll look at the results, and we hope to improve the ability to measure memory and thinking ability. And so you will, maybe in the future, won't have to come in for that hour and a half or 2 hour testing annually if we can find that this helps. So it is all very cleverly designed by Dr. Hassenstab, 7-day week. You might be tested at different times for each day. When you're prompted to test and you're driving your car, you can delay the testing. You don't have to stop and pull over. You can wait for a couple hours to do it. But each test will be, as I said, less than 3 minutes, so hopefully non-intrusive. And Jason has thought of some ways to encourage people to do this, including, and pretty generous, if you complete one of those 3-minute tests, you get 50 cents. But it adds up over the week and over the year. So hopefully, this is very innovative. This is really terrific, but it may be the wave of the future. All right, so these are the, I won't go through the specifics, but these are the leaders of the Knight ADRC and the Memory and Aging Project. And we try for diversity, as I said, and we're quite aware that most of our leaders are Caucasian, 2 are Hispanic. We don't have any African American leaders, but we do have gender equity. We have a total of 8 leaders who are men and 8 who are women, but we are always looking to be more diverse. Now, I turned 71 in February. Unlike Dr. Buckles, I don't have plans to retire, but I don't also have plans to work forever. So at some point, I am going to retire. As I say, I don't have plans for it. And I've asked all of our leaders, myself included, to develop a succession plan. Who, if they have to leave or they can't work, who would they want to take over for them? And so I've been thinking about a succession plan, and I've hit upon 2 individuals. I'm reading to my 2 grandsons, Jack and Thomas, the Knight ADRC manual, so how they can find out how participants are doing in terms of their memory. So as soon as they're ready, then maybe I will retire. So with that, let me ask Dr. Cummings-Vaughn to come to the podium, and she's going to talk about looking at how we assess memory problems and whether race plays a role. Let's welcome Dr. Cummings-Vaughn.
Good morning. So Dr. Morris really set up my talk quite a bit by really explaining the importance of making sure that we're fully investigating the causes and treatments of Alzheimer's dementia in all people, so that we can really help the full population of people, especially those in St. Louis, since we're here. So my study that I'm going to be explaining is about self-report and informant report. And in terms of the testing, when all of you have come in, you've been asked these questions about what you think about your memory and thinking. Do you feel like there's been a problem? And these things are actually quite important. Noticing your own medical problems or how you're doing is important. And then having healthcare professionals listen to you is also very important. So I think this is good information for everyone that's here. So as Dr. Morris said, early detection is really important. And looking at this self-report and informant report, we can get an idea how early we're able to find out when people are having problems with memory and thinking. And is there a difference in terms of race in detecting whether or not people are having problems? So is it that we're not noticing it as much in different individuals? Is it that we're not being listened to by our healthcare professionals? In terms of all healthcare, but specifically Alzheimer's disease, there are differences in terms of there are disparities by race in these disease processes. And is it purely genetic? We talked a little bit about allelic or genetic differences, the expression of the apolipoprotein E in different individuals. So are those differences very significant between different races? Is it social determinants of health? So medical problems that are brought on by where you live and how your life is led that really contributes to different people's risks? Is there just some difference in the prevalence of other associated diseases like diabetes, coronary artery disease? Are there differences in terms of stress in life and other social factors that might be affecting these disparities? I already talked about lack of detection. So is that an important factor in the differences we see, the racial differences? And is it lack of access for evaluation and treatment? So we know that lack of access for healthcare in general is a major difference between different races throughout the United States. And is this a major factor in terms of Alzheimer's disease detection? And will this be important when we think about treatment? So there's lots of studies about perceptions and other differences between African Americans and Caucasians. And I'm concentrating on African Americans and Caucasians only because these are kind of some of the largest groups that we have represented in our studies. But I think differences throughout all races is very important for us to look at. So in this study that was performed in 2013, there were some differences in the perception of Alzheimer's disease or dementia. So I think perception is incredibly important. So if you feel that changes in memory and thinking is just something that happens naturally with age, you're less likely to report that to your healthcare provider. And if you're less likely to report it, you're also less likely to be followed, less likely to receive treatment for it. And so this can lead to a lot of different problems. So it's important for us to find out if there are differences in perception, provide education. So in this study, they did find that there were differences in perception. Both African Americans and Latino respondents were more likely to perceive stress as a potential risk factor. And so I think whether it's stress or feeling like it's just a natural part of aging, this could contribute to the disparities that we see in terms of detection and prevalence of disease process. So there have been other studies performed on the cohorts that Dr. Morris talked about, the 3 cohorts that has the adult child study and the memory and aging project groups. And so we looked at, or my colleague, Dr. Carr, looked at the differences in self-report and informant report and the whether or not it predicted future Alzheimer's disease. And this was kind of 156 individuals, and there were differences. So informants were very good at distinguishing memory problems and predicting future disease. So I think this is important because what you see in your loved ones is important. Accompanying your loved ones to their medical appointments and expressing this to their healthcare provider is important. But are there racial differences? And so this is what I looked at. So there were a total of 1,169 participants that we looked at their data that they contributed since 1985. There were 163 African Americans and 1,006 Caucasians. You can see these numbers might seem confusing, but these are every single time a person was assessed. So that's why the numbers are 4,000, 2,000, because every single year you're coming in and doing an assessment and being asked whether or not there have been changes in a person's memory and thinking. How do you feel about the memory and thinking, both you and your loved one? And when that happens, we as clinicians we do a clinical dementia rating. And this represents our feeling about our assessment of your memory and thinking based on the history that's given. And so from this data, we found that in terms of Caucasians, 49% remained normal over the years from 1985, anytime from starting the study until we ended looking at all of the data. 34 progressed into some sort of clinical dementia. 21% of African Americans remain normal, and 72% progressed. So there's a significant difference. And keep in mind the significant difference in numbers. There's only 163 African Americans and 1,006 Caucasians. And we can talk later, you can think about why that would be, but obviously you all are wonderful. We're choosing for people who are interested in participating in research, who might have a concern about their own memory and thinking. So I think this has an impact. But despite that, I think it's really important. This is a statistical difference that really we should look further into. So when we looked at the responses and progression of individuals as when compared to what participants and their informants thought was going on with them, African American participants who self-reported problems with memory and thinking were 3.5 times more likely to convert to clinical memory loss. Caucasian participants who self-reported were 2.3 times more likely. So from my standpoint, I think that means that you do realize what is happening to you often, and it's important for all of us to look at it. But I think it's also interesting to see that, especially for African Americans involved in this study, they are more likely than their Caucasian participants to notice these differences. So in terms of informants, as I said in the previous study, informants really had a much better idea about changes happening than participants. Is that true? And does that hold true in terms of differences by race? And we find that there's a very significant difference between races, but you can see 8.5 times more likely to convert to clinical memory loss if an African American informant feels that the participant is having problems with their memory and thinking. I think on many levels that's very interesting, and we hope to look more into that. So overall, African Americans and Caucasian informants are better able to discern progression than participants. And this is not necessarily surprising, but I think for participants, you still have an idea of what is happening with you. And I think it's important for you to speak with your family members and caregivers about what is happening. African American participants and informants are better than Caucasians at noticing problems that are leading to clinical memory loss. So again, what does this all mean? I'm actually not completely sure, but I do think it's significant, and I think that we should look into this, because anything that is leading to differences in detection and potentially progression of disease are things that we really need to look into. So again, I already said that all of you are interested in being study participants or interested in memory loss. So that kind of biases the sample versus people out in the community. The number of African American participants was much lower, which is why we are very interested in recruiting as many African Americans as possible into the study. And then there are significant this difference in the percentage of individuals who are converting to clinical memory loss. What does that really mean? And we have yet to look at all of the supporting data. So all of this tissue and biomarker data that you have provided, we haven't compared that in terms of the informants, and that's part of what we plan on doing. So thank you very much.
Thank you very much, Lenise. So now I think everyone understands why we ask not only you, the participant, to come to our assessments, but also someone who knows you well, the study partner, because study partner is very sensitive. Dr. Justin Long, just 1 year ago, completed his residency training as a neurologist and decided to do his practice and research in Alzheimer's disease. So he has a laboratory project in Dr. Holtzman's laboratory, but also is working in the Memory and Aging Project, and he's going to report on whether infections can play a role in Alzheimer's. Let's welcome Dr. Long.
Thank you, Dr. Morris, for inviting me to speak today on this topic. As Dr. Morris mentioned earlier, this is a bit of a hot topic, and one reason that I was asked to present on this is in the last year or so there have been... Is that better? Okay. So in the last year there have been several high-profile studies that have made it into the press that some of you may have seen, and so we thought it was important to briefly review some of that data and put it into context so that you might be able to better interpret what you're reading in the press. And so I'm going to be talking specifically about whether infections might contribute to Alzheimer's risk. So I'll start off by just stating that there are some forms of dementia that we know are definitively caused by infection. So as an example, syphilis that is untreated for many years can lead to a syndrome called general paresis, which is associated with memory loss and psychosis and other neurologic symptoms. Similarly, HIV, if it goes untreated for many years, can lead to a form of dementia called HIV-associated dementia. Herpes simplex virus, which is the virus that causes cold sores on your mouth, can infect the brain, causing encephalitis, and in some cases can lead to permanent brain damage, leading to a dementia syndrome. Tuberculosis is a bacterial infection that in rare cases can infect the brain and not cause overt symptoms and instead present with slow decline in memory. And then there are protein particles called prions that we know in very, very rare cases can transmit between humans and can cause a form of a rapidly progressive dementia. But these are all very rare, and we rarely see these in neurology clinics or in the neurology wards in the hospital. So the question is, what about Alzheimer's disease? And so as I mentioned in the last year, you might have seen several news articles. In the New York Times article, common virus may play a role in Alzheimer's disease. Newsweek, dementia and gum disease, Alzheimer's linked to gingivitis. NPR, story on infectious theory of Alzheimer's disease, and CNN also with a story discussing gum infection and its link to Alzheimer's disease. So the question is, what does the data say? And so I'm going to review briefly some of the data that was discussed in some of these recent studies and put it in context with some other research that's been done over the last 20 years or so. So briefly, one of the hallmark proteins of Alzheimer's disease is the amyloid beta protein, as Dr. Morris mentioned earlier. And there's some evidence that amyloid beta may protect against certain types of infection. So if you add amyloid beta in a test tube to various bacteria, yeast, or viruses like the herpes simplex virus, it can actually entangle and trap those pathogens and limit their ability to infect cells. And there are certain mouse models of Alzheimer's disease that have been engineered to produce high levels of this amyloid beta protein. And if you directly infect these mouse models with bacteria, yeast, or various viruses, they don't die as quickly. They live longer. So this is some evidence that amyloid beta may actually protect against some certain types of infection in these experimental animal models. This is a picture of one of these animal models, and one of these recent studies showed that if they experimentally infected these mice that generate a lot of amyloid beta in their brains, that the areas that had herpes simplex virus deposition also accumulated the amyloid beta protein. So this is somewhat similar to what we see in the brain of Alzheimer's patients. And so this was potential evidence that maybe infection might promote accumulation of amyloid. And then another recent study looked at infecting these same types of mice with periodontal bacteria. This is bacteria that causes gum infections, and found that mice that were infected with this bacteria developed increased amyloid in the brain in a similar way to what we see in Alzheimer's. Now that being said, these are animal models, and no animal model really perfectly reproduces what we see in humans. And so if you really want to understand whether infection is a risk factor for Alzheimer's, you really need to look at data in humans. So what does that data say? So there is some data that gum infections, periodontal infections, participants or patients who have higher rates of periodontal infections might have an increased risk of memory decline. And there was one study last year that showed that these periodontal bacteria proteins were present in the brains of Alzheimer's patients, but not in patients who had no Alzheimer's disease pathology in their brain. There have been some studies that have identified the herpes virus in the brains of Alzheimer's patients and found that this virus might be present in the plaques that accumulate. And then there have been some studies that have looked at the risk of developing dementia and memory decline and associated that with in various infections, including virus, and found that there might be a slight increase in the risk of dementia after herpes virus infections, including the virus that causes shingles. Now, that being said, these studies really are all over the place. There are studies that have shown no increased risk, and others that have shown a very small amount of increased risk. In medicine, when we have a lot of studies that show different results, we like to look for something called a meta-analysis, where they combine the results from multiple studies and try to summarize them and see if we can gain greater insights. And there was a recent meta-analysis looking at the risk of dementia after herpes virus infection, and one of the conclusions was that although herpes virus infection may be associated with dementia, the results were inconsistent across studies, and the overall evidence was very low quality. So even this high-level study couldn't definitively make a conclusion. So what's the takeaway from these recent studies? And I'll just say that reasonable physicians and scientists might come to different conclusions. This is my interpretation of the data, and it's just something that you may be able to take away yourself. But there may be others who have come to different conclusions. Overall, I think the human data associating infection with risk of dementia is weak. And there hasn't been a single study that's demonstrated that infections definitively cause Alzheimer's disease in humans. These are just associations. People with infection might also have a higher risk of developing Alzheimer's disease. And there's no evidence that a single infection might trigger Alzheimer's disease. So I don't think you should leave worried that if you develop a shingles infection, you're at a much higher risk of developing Alzheimer's disease in the coming months. And there's only very limited evidence that if you treat infection, such as herpes virus infections, that this reduces your risk of developing Alzheimer's disease. So certainly there's no evidence that you should undergo a different course of treatment if you do develop a bacterial or viral infection. And if these do play a role, they are likely but one risk factor among many. I'll just emphasize that age is still the strongest risk factor, and the risk due to infection would be much smaller. And overall, we just need further longitudinal studies looking at how people change in terms of their memory and thinking after developing an infection over time, and that will give us better evidence down the road as to whether risk of infection actually increases risk of dementia. So with that, I would just like to thank all of you, all of our participants, for enabling our studies. As Dr. Morris mentioned, I am the Knight ADRC Fellow, and so I'd like to thank him as my clinical mentor and the director of my fellowship, and Dr. Holtzman, who is my research mentor. And I also receive funding support from the Alzheimer's Association, so I'd like to thank them as well. Thank you.
So thanks so much, Justin. Even though Dr. Long says the evidence for infection causing Alzheimer's disease in humans is weak, please don't get sick, okay? If you have questions, remember to write them down on the card. We'll pick them up at the end of the talks. And now it's a pleasure to introduce straight, direct from Lambert Airport, Dr. David Holtzman.
Well, good morning, everybody. It's great to be here, be back home. And I want to first just thank everybody for participating in the Memory and Aging Project. This is, you're really, really contributing to the worldwide effort to treat and cure Alzheimer's disease. So thank you so much. And before I start, I did need to say, being from St. Louis originally, that it's pretty amazing to say on June the 1st, let's go Blues. So what I wanted to do today is just give you an example of how different treatments that might prevent or treat Alzheimer's disease move into patients. So what I'll do is give you an example from my own laboratory of something that is now being used to try to treat patients with Alzheimer's disease. And then I'll update you with some of the trials that Dr. Joy Snyder and others are leading here at the Knight ADRC. So as a faculty member at Washington University, we are required, if we have any potential conflicts of interest, to disclose them. And so because the treatment I'm going to tell you about, the story, was developed in my lab, in order to move it into clinical trials, we needed to license it to a company. So there's financial interest that both I have as well as Washington University if this moves forward further. Hopefully that's a good thing. So as all of you have been hearing about over the years, the two major neuropathologies that underlie a lot of the damage in the brain in Alzheimer's disease are called plaques and tangles. So the plaques are made up of the amyloid beta protein, which accumulates outside the cells in the brain. And the tangles are made up of the tau protein. And this is a protein that accumulates, builds up in the brain inside nerve cells and leads to damage of the nerve cells. In addition to that, wherever you see somebody is failing in memory or thinking, those parts of the brain degenerate. So those are the major features of the disease. And the other thing that's recognized a lot more in the last few years is that there's inflammation in the brain where these lesions occur, and that inflammation is also likely contributing to the dementia and cognitive decline. So you probably have seen different clinicians and scientists show you curves like the one I'm showing you on this slide. So what this shows that in somebody who develops the dementia from Alzheimer's disease, what probably happens is about 20 years before the symptoms begin, this protein called amyloid beta begins to accumulate in the brain. And you can see that it's this red line. You can see the slow accumulation over time. That happens while people are still cognitively normal. And then sometime right before people become cognitively impaired, the tau protein starts to accumulate in the brain. And what's not shown on here is inflammation is also occurring during these different time points. So in terms of the treatments that have been developed for Alzheimer's disease, there are, many of you are probably aware, there are symptomatic treatments that help a little bit attention and memory. So like cholinesterase inhibitors and memantine. Those are typically given to people who at least have very mild or moderate dementia. And you can see the time point, 30 years after people have been building up these lesions in the brain is when we give these current treatments to people. One of the things that the whole field has been working on for some time is that we want to try to get rid of these amyloid plaques that build up. So to clear plaques from the brain, I ultimately think will be a treatment for Alzheimer's disease, but the question is when would you want to do that? Of course, you'd probably want to do it before it starts so that you prevent it in the first place. But so far, what's been tried is that there's been types of drugs called antibodies that have been developed that can now clear these plaques from the brain. But so far, there hasn't been success in using them in people that already have dementia. One of the other things that is still very much in early development is to try to prevent the buildup and spreading of this tau pathology or tangle pathology in the brain. Because the evidence suggests that the buildup of that protein is really the thing that's damaging the brain. And so I'll tell you an example of that in a moment. There are some trials that are ongoing where we're trying to clear the amyloid and prevent the tau spreading in the brain, as well as treating inflammation. I'll tell you a little bit about some of those studies that are ongoing that Dr. Snyder and others are leading here. And then ultimately, we'd like to really prevent this whole pathology from beginning in the first place. And Eric McDade is about to start a study here, a worldwide study, to try to do this in dominantly inherited Alzheimer's disease. Okay, so I'll tell you the local story that occurred in our group over the last few years. So Alzheimer's disease and other diseases like it often are called proteopathies, meaning that proteins in the brain that normally are soluble, they float around, they clump up, and they build up in the brain. And one of those proteins that does that is tau. And it can accumulate in different diseases in nerve cells or the supporting cells, the glial cells. So there's different types of diseases where tau accumulates: Alzheimer's, frontotemporal dementia, progressive supranuclear palsy. These are all different tauopathies. You may have heard in the National Football League and in other sports, people that have repetitive brain injuries have developed a tauopathy called chronic traumatic encephalopathy. So that's another form where disease where tau builds up in the brain. And in this picture on the bottom, you can see in a brain of somebody that ultimately develops Alzheimer's disease, tau begins to build up in the brain in a part of the brain called the hippocampus. And then it somehow spreads from that region to other regions in the brain. And when that happens, that's when people begin to show cognitive abnormalities. So one of the theories as to how this works is that the tau protein itself is moving between cells and it's spreading. So that's illustrated in this next slide. So what one possibility, we think from animal model experiments, that you can see this blue material is tau that's clumped up in a nerve cell. And one of the things we think happens is that it exits the nerve cell, gets into adjacent or connected nerve cells, and it corrupts the tau in that cell, causing a spreading or seeding and spreading phenomenon so that it moves throughout the brain. Well, if that happens, and there's evidence certainly in animal models that this happens, that you could maybe catch it and prevent that from happening. And one way to potentially prevent that are with antibodies. These are molecules I'll talk about in the next slide that can capture other proteins and cause them to be degraded. So these antibodies are large proteins made by our immune cells called plasma cells that are in our blood. And what we do normally is we use antibodies to fight infections. However, we can also engineer antibodies to actually do things like fight cancer. Some of the newest treatments to treat what was previously untreatable forms of cancer are antibodies. But they can also do things like remove amyloid from the brain. And we think they might be able to prevent this tau spreading in the brain. So, oh gosh, it was probably already about 10 years ago, my laboratory was beginning to study the tau protein. And in order to do this, we developed a series of antibodies to the tau protein. And once we started using these antibodies, one of the things we did was to try to figure out if they actually blocked this tau spreading from cell to cell. And we found several of them that could do that in a test tube. So then we went on to test whether in an animal model that develops tau pathology, whether any of these antibodies might decrease the tau pathology in the brain of an animal. And what's shown in this slide is an animal on the left that develops these neurofibrillary tangle pathologies. And all of that brown staining is abnormal tau that's building up in the brain of an animal. So what we did in this animal is that right when this buildup was beginning, we gave it an antibody to the tau protein. And we had, in this particular case, we administered it in the central nervous system for 3 months. And you can see in the animal on the right, which received the antibody, it developed much less tau pathology in the brain. So we then went on to test whether or not if we gave an antibody like this in the vein, or in the veins of animals like you might give to a human, every week for 3 months, whether it would also have a similar effect. And what you can see in this slide is that in the animals on the left, they were given placebo, and the animals on the right, they were given an antibody. And there was also, again, some decrease of this tau pathology that we saw. We also asked whether the brain was any better, other than having this pathology, whether the brain was not shrinking as much. Because we know in Alzheimer's disease, for example, as you develop the disease, the brain loses cells and it starts to shrink. And what you can see on the right are this white bar is a group of animals given placebo, and we're measuring the volume of part of the brain. And the animals given the antibody had about a 25% bigger brain in that region. So it suggests that not only was the antibody decreasing the tau pathology, but also protecting the brain. So since this antibody was promising, in order to actually develop it and move it potentially into human trials, it needed to be humanized. What that means is that in mice, like humans, make antibodies, but the sequences are slightly different. So in order for a human to be able to tolerate getting an antibody, it has to have human sequences in part of the protein. So to do this, we licensed the antibody to a company called C2N Diagnostics. And then the company, with financial support from a foundation called the Rainwater Foundation, developed the antibody to do what's called a phase 1 clinical trial. And that's where you actually give the antibody to people to see if it's safe and whether it actually interacts with its target, tau protein. So what you can see here is when we give this antibody to mice on the left, you can see these bars represent tau increasing in the bloodstream of the mice. And we think this happens because the tau protein, while it's in the brain, some of it gets out into the blood, and this just shows that the antibody can actually bind the protein target in the blood. And if you look on the right, in the first phase 1 clinical trial in humans, you can see the same phenomenon. So you can see these bars rising. These are different humans that were given the antibody, and also you're now seeing this tau protein in the blood because the antibody's capturing it. Just means it can bind what is intended to bind. What we think it needs to do, though, to really treat the disease, some of the antibodies get in the brain, and it's probably binding the tau in the brain. And that's probably why we were able to see previously that it had some effects in mice. So what's going on now is that following completion of this phase 1 trial, the anti-tau antibody was licensed to a large pharmaceutical company called AbbVie. And the reason for this is that these trials, once you get into larger numbers of patients, are very, very expensive. And the only people that can really afford to actually run these trials are companies that actually have a lot of resources to do that. So the anti-tau antibody, it happens to be called ABBV, which is AbbVie, and it just has a number right now, AD12, is now in 2 large phase 2 clinical trials. So phase 2 clinical trials are where you're beginning to test whether the drug, or in this case antibody, will actually begin to show effects. So there's a 1 year or 12-month trial that's ongoing right now of about almost 400 people in a disease called progressive supranuclear palsy. That's a disease where tau builds up in the brain and people develop kind of a Parkinson's-like syndrome where they have trouble moving and they have trouble with their balance. That's completely enrolled, and the results of that study we'll know in about 8 months from now. In a second, almost 2-year trial, 96 weeks, there's 400 to 450 people with very mild dementia due to Alzheimer's disease that are also in a trial with this antibody. Both trials are completely enrolled. So the Alzheimer trial will read out in, I think, about 1.5 years or so from now. So that's where that just gives you an example of how a drug, there's many drugs that are like this that have been developed over time that have moved into trials, how it moves from a laboratory to initial testing and then into people. So what's going on right now at the Knight ADRC? And these are predominantly studies that Joy Snyder is, clinical trials Joy is leading. One is called the Engage Emerge trial. That's no longer enrolling. That was a phase 3, so a really large trial worldwide that was studying a drug that you may have heard of in the news recently called aducanumab. This was developed by a company called Biogen. It was very promising in that it can remove amyloid from the brain. We enrolled 10 people in this trial of 1,600 worldwide that were in this, and the study was unfortunately stopped due to what's called futility, meaning before everybody finished the trial, they looked at some of the information, and there must not have been evidence that the people were getting better compared to the placebos. This was very unfortunate. There's many reasons for why this may not have worked, but the data still has not been released, so we don't really know how to interpret this yet. But we are still completing safety visits. There's also a study called Trailblazer. This is no longer enrolling. It's already completed enrollment. It's also a phase 3 study by Eli Lilly with an antibody that also binds amyloid, and that's moving forward, and that will hopefully go to completion, and we'll find out in about a year or two the results of that trial. There's clinical trials that are currently enrolling in people with symptomatic Alzheimer's disease in the early stages. Dr. Lucy, who's here today, is running a trial called Discover. That's a phase 1/2, our early trial of a drug called Posifen that may reduce amyloid beta in the body. And you can read more about that in your handout. In the dominantly inherited Alzheimer network trials unit, there are 2 treatment arms that Randy Bateman is leading with many people. There are 2 drugs in that trial now, 2 antibodies, and there will be a new arm, I'm sure, that will start in the next year. The A4 study is a very exciting study that is now fully enrolled nationwide. This was the first prevention trial in people with who potentially are at risk for late onset Alzheimer's disease. And this is with an antibody against amyloid beta called solanezumab or placebo. It's the first prevention study in people with sporadic Alzheimer's disease, as I just mentioned. Over 1,000 people are enrolled, and the treatment period will end in July of 2022, and results soon available after that. So it's still a little while away, but everybody's now been enrolled in the trial. And Joy, if I'm not mistaken, is almost a 5-year treatment, or 4.5 years, something like that. Okay. And then what's kind of come next at our center? One study that is starting is called Graduate. It's another large phase 3 study with an antibody called gantenerumab. It also removes amyloid from the brain. It will be done in people with mild Alzheimer's disease. We'll enroll about 6 to 10 people. So hopefully many of you'll be hearing more about this as we try to find participants. There's Clarity. It was a phase 2 study with an antibody called BAN2401. It's also an amyloid antibody, but it has a little bit different features to it than some of the other ones. That will enroll 6 to 10 people. And then there's another prevention trial that will start in the next year or two called A45 and A3. So these, again, are prevention studies for people at risk for Alzheimer's disease, but still are cognitively normal. And we'll be probably using this antibody BAN201 as well as a drug, a BACE inhibitor, which decreases the production of amyloid. So I think that is it for the slides. So I want to thank you all again for being here, and thanks.
Thanks so much, Dave. Now you're going to hear 4 very short presentations of studies that are either started or will start soon, or will lead to new avenues of pursuit. The first is Falling, and that's going to be presented by Susan Stark, who is in the program of occupational therapy. She's collaborating with Bo Ansis from neurology. Let's welcome Dr. Stark.
Good morning, everyone. Thank you so much for coming today. Dr. Morris talks about how amazing our research team is, but we would be nothing without you, and we are so grateful for your generosity. I am delighted on behalf of Dr. Ansis and our research team today to tell you a little bit about our work with preclinical Alzheimer's disease and falls. So it's no surprise to you that falls are very common. We know that on average, 1 in 4 adults age 65 or older will fall in the next year. Many of you are not average. In fact, most of you are not average. And your individual fall risk is based on your individual fall risk profile. So the number of fall risk factors that you have, like maybe limitations in your ability to walk or move, or diminished strength, or super strength, or difficulty seeing, those are the things that increase your fall risk. If you have no risk factors, you have about a 10% chance of falling in the next year. If you have 4 or more, that risk goes up to 80. One of the really interesting risk factors that I've been intrigued by is that if you have Alzheimer's disease, actually over 3 of 4 older adults are going to fall. So it increases your risk to 60 to 80% chance of falling in the next year. The consequences of falls are serious. It's the #1 cause of brain injury, the #1 cause of hip fracture, the #1 reason that people have to go into nursing homes prematurely. This comes at great cost to us in our society, about $50 billion a year annually. And the most difficult thing for me is it's the #1 cause of premature death for older adults. We do know that if the fall rates continue in the United States, by the year 2030, the CDC predicts that we'll have 7 fall deaths every year. The good news is falls are preventable. We have lots of effective treatments that we can deliver based on individual risk factors. One of the things I'm interested in, though, is a risk factor we don't know much about, and that's the relationship between falls and preclinical Alzheimer's disease. So some years ago, many of you helped us, and we began to understand that people who have preclinical Alzheimer's disease have actually a 3 in 4 risk, or a 75% risk of falling. So these people with preclinical Alzheimer's disease look more like people with dementia, with Alzheimer's disease, than the general population. We don't know why, but we know that that relationship exists. So this year we were fortunate to convince the National Institute on Aging to give us some more funding to begin to explore this issue to understand why this relationship exists. So we are once again partnering with the Knight ADRC participants, and we're looking for folks who are 65 or older, who are CDR 0. And what we would like to do is send an occupational therapist into your home to conduct an individual fall risk assessment. So these OTs will come in, they will conduct a battery of assessments. We will use that important data with what you've already contributed to the Knight ADRC, all of your biomarker data and clinical assessments, to begin to try to understand why there's a relationship between preclinical Alzheimer's disease and falls. So we want to leave you with something really valuable. So if you participate in our study, those therapists will actually give you all of your research results. So you will have an individual fall risk profile that we hope you'll share with your doctor. In addition, these experts in fall prevention will give you access to resources in the St. Louis area that will help you reduce your individual fall risk. And as always, in our research study, we'll check in with you on a monthly basis to see if you've had a fall. And you'll get that $5 gift card for those of you who were in our previous study before every time you take the time to answer an email or a phone call. So today, at the back of the room, let's see, I think we have Becky and Regina, and we have 2 of our other occupational therapists. There they are over there, they're waving. And they have a table set up. And if you would like to come and learn a little bit more about our study, we have some of our equipment, some of our balance boards and the ways that we'll test you, and we would love to answer any questions that you have about our study. Thank you. Oh, one thing, we have about 200 more spots over the next year for people that are interested, so let us know. Thank you.
We were talking about Dr. Hassenstav's online smartphone assessment of cognition. Here's another opportunity to look at online assessment. Dr. Buckles is going to present it. Welcome, Virginia Buckles.
Thank you all, and I'd like to add my thanks for the last 27 years I've been at these meetings and have appreciated your contribution every single year. Thank you. So we're collaborating with a team at the University of California, San Francisco, who has created an online brain health registry. And they've asked us to collaborate with them to develop a online version of some of the things we do in the Memory and Aging Project. So this online validation of clinical measures to detect cognitive decline is the title. Or, is your computer as good as a map clinician? So this is maybe a little threatening to some of us, but so the aims of this study are to evaluate whether the judgment of a skilled clinician, which is the gold standard for evaluating cognitive aging, can be replaced by a computer-administered and scored assessment that we can then compare with the clinician judgment. So this will require administering the same tests, or nearly the same tests, both in the Memory and Aging Project when you come for your annual visit, and then at home on your computer, so that we can compare the two assessments. So both the participant and the collateral source, the study partner, you're not getting out of it that easy, will have to be involved in this. And we will use that existing brain health registry data collection tool to collect this information online with the versions of what we do in the Memory and Aging Project. You'll be asked to register with that online system, and some of their tests will be administered as well as our own tests. So annually, you will complete your usual MAP visit, plus online testing at your MAP visit. So you'll be getting clinically evaluated and then taken to another room where there's a computer to do the online testing. And then within a few weeks, you will do another online testing. It could be before your clinical visit or after your clinical visit, and then 6 months later. So that testing will last for 2 to 4 years, depending on when you enroll in the study. So the online testing time commitment, again, so you're testing 3 times a year online, 1 in the lab, 1 in the clinic, and 2 at home. And for the participant, this takes about 80 minutes. And you can see here are the tests that are collected. And for the study partner, or the collateral source, it takes about 60 minutes. So what are the goals of this? So at the end of the study, we will compare the results from the Memory and Aging Project with what was collected online. All right, so we know pretty much, and we're not threatened, that these online measurements will not be as good as what is collected in the Memory and Aging Project. So we're pretty sure about that. But so why do this? What's the point? Why would we commit to this? So MAP can only assess so many participants per year. I think Dr. Morris said that was around 900. With an online registry, that number can be increased to tens of thousands of people. And so we get a much broader reach from this online measure. And if it can be close to what the quality is of what we collect in the clinic, this would be a good thing. It can help with other types of research, it can help with screening, it can help with clinical trials. It could possibly reduce the costs of doing the research compared to coming in and seeing a physician and nurses and social workers in the clinic. And finally, it's possible that this could reduce the burden on the participants as well as the cost in the clinic, the amount of time to visit the clinic, etc. So we will be starting to recruit for this study in a little bit after I retire. And so I won't be part of this, but there will be a Memory and Aging Project coordinator who will start this project. So, thank you.
Okay, we're interested in the role of hearing and whether that can contribute to or be affected by Alzheimer's disease. And Mitch Summers from our Department of Psychological and Brain Sciences is going to talk about a new hearing study. Mitch?
Good morning, and let me add my thanks to all of you for your willingness to participate, for your willingness to sit here on a nice Saturday afternoon, or Saturday morning rather. We really couldn't do this research without you, and so really our sincere thanks for all your contributions. We are interested in studying hearing, and in particular the relationship between hearing and speech perception. And let me start off by asking you two questions. So in the last year, how many of you have found yourself at some point having difficulty understanding a conversation in a crowded room, maybe such as this, maybe even this morning? If you could just give a show of hands, how many people? Virtually everybody. Now let me ask you a second question. In the last year, how many of you have had your hearing tested? A good amount, but certainly less. And so one of the things that we're concerned about in studies of hearing is that it's one of the most undiagnosed or difficult, not difficult, but less underdiagnosed disorders that individuals, both normal, healthy older adults and individuals with Alzheimer's disease have. And so we've been interested in the relationship between hearing and to some extent cognition, because we know that about 70% of individuals who are 65 or over have difficulty with hearing, and that there are really some significant consequences to this. One of the things, for example, is social isolation. Often people who do have difficulty understanding speech or conversations avoid situations like parties, like restaurants, like family gatherings, where there's other noise in the background, and it can be very difficult to understand what's going on. And so we want to make sure that, and look at the relationship between, again, social isolation and hearing loss. And there is some exciting and interesting new evidence to suggest that there may be a relationship between hearing loss and cognitive impairment. So in our study, we want to provide a comprehensive test of hearing and speech perception. So if you come and enroll in our study, you'll have 2 sessions where you'll get an extensive audiological evaluation, including measures of hearing, speech perception, and we'll even measure a little bit of how well you can produce speech. We've tested about 17 participants so far. Every single one of the participants that we have tested has had hearing loss, and we've been able to refer almost 9 of those to audiological services, and we've even had one who has contacted us again and indicated that because of the study, they were able to get a hearing aid and have significantly improved their ability to understand speech. So in addition, we have some general questions that we're interested in. So as I said, one of the exciting areas is there a relationship between hearing loss and cognitive impairment? Is it the fact that if you have a hearing loss, you have reduced sort of input to the brain? Does that contribute to some of the deficits that we see when people who have Alzheimer's disease? Can we use measures of hearing and speech perception to maybe predict who is and who isn't going to develop Alzheimer's disease? One of the things in terms of adding to our ability to diagnose Alzheimer's disease early on. And in particular, we're also interested in what are some of the best ways to both test people with Alzheimer's disease in audiological evaluations. Some of them can be relatively lengthy. Some of them can be somewhat challenging for individuals with difficulty with memory. And so we wanted to devise ways to develop and test people who do have Alzheimer's disease and to provide those audiological services. And how can we treat individuals with Alzheimer's disease? What are some of the best treatments? Relatively little information about there is how Alzheimer's disease affects our ability to treat individuals with hearing loss. So we'd be happy to answer any questions. We'll have flyers in the back about participating. And thank you very much for your time and for your participation.
Thanks so much, Mitch. So the key deficit of Alzheimer's dementia is memory loss. We have also from the Department of Psychological and Brain Sciences an investigator who's very interested in how memories are made and how one can benefit from memories, Dr. Jeff Zock. So let's welcome Jeff.
Thanks, Dr. Morris. And let me just add my thanks to all of you. I've been working with participants from the Alzheimer's Disease Research Center for more than 15 years now, and it's been personally and scientifically one of the most rewarding things that I've done. So my lab is a cognitive neuroscience lab. I'm right next door to Dr. Summers over in the newly renamed Summers building, formerly known as the Psychology Building, but it's Summers with only one M, so you got to keep those straight. And in the lab, what we do, we're a basic science lab, but we're very interested in how basic processes of perception and comprehension and memory and learning relate to healthy aging and Alzheimer's disease. And so what we do is try to collect data on behavior and brain activity, use that to build computer models that then can drive tools for assessment and for treatment. And one of the things that we've been really struck by over the last few years is the question of why we have memory at all. If you look at what we do in the lab in the clinic, you would think that the reason that humans and other species evolved multiple systems for memory in the brain that are shared, many of them widely across species, was to sit in a chair and think about word lists that someone gave us recently. We think that's probably not why memory evolved. Rather, we evolved memory systems because they allow us to use experiences from the recent past to predict what's going to happen in a new situation. So when you drove up here this morning, if you had been over to Westport recently, you were able to use that experience to make predictions about what's going to happen if you turn left versus right and where the open parking spaces are likely to be and so forth. This becomes particularly an acute concern when we encounter changes in our environment. So if you hit a confusing detour, that your predictions based on your recent memories are immediately going to be pointing in the wrong direction, and you've got to somehow solve this problem. If you can use your memory to drive those predictions and then use it to update your representation of that environment, that can be beneficial in the longer term. So what we've learned over the last couple of years in collaboration with you all is that indeed people who are able to use their memory for recent episodes are able to improve their performance by better predicting what's going to happen in a given situation. When things change, this can lead to errors in the short term, but it can lead to improved learning for the next time. Healthy aging is associated in changes in these mechanisms. So one thing that we see is that with healthy aging, there's a reduced use of memory for the recent past to guide predictions, and a reduction in learning from these prediction errors. What we would like to learn going forward is how this works in the brain. So the study that we have just concluded in collaboration with you all, and some of you I know participated, and thank you for it, involved watching movies that contained repeating experiences and changing experiences during MRI scanning. And this is going to help us understand the mechanisms by which this learning from changes happens. And then how we can give these processes a helping hand. So hopefully come fall, we'll be enrolling for a new study in which we're going to be intervening on these memory encoding systems to see if we can produce better predictions and better subsequent memories. I'm very excited about these studies, and I look forward to seeing y'all in the lab. Let me say a little bit about studies in the lab. So we are called the Dynamic Cognition Lab. We're located over in the Summers Family Building on the Danforth campus. And one thing that's kind of uniquely characteristic of studies in our lab is that rather than studying artificial materials of the type that we often use in the lab in the clinic, if you've been in one of our studies, you know that mostly what we do is show movies and have people read stories. So good times all around. We also do a lot of studies with undergraduate participants, and they universally report, our participants of all ages, that compared to some of the other things that we ask people to do, it's a pleasant experience. Sessions can take 1 to 2.5 hours, and I hope to see you in the lab. Thank you. Will there be popcorn? Not in any of our studies to date, but that's not a bad idea.
So thanks very much, Jeff. I want all the speakers to please come up here and have a seat. We're going to collect your questions that you're writing down on your cards. Memory and Aging Project staff will be coming around, so hold yours up. While they're being collected, while our speakers are coming to the table, you know that some of you may recognize this, we give recognition pins to our participants who have been in the study for 5-year intervals. So we have a 5-year, 10-year, 15-year, 25 years recognition pins. I mentioned at the outset that Ted has been with us for 32 years. We don't have a 30-year pin, except now. This is your 30-year pin. So let's welcome Ted. So all the speakers are here. Let's get going. Let's see. Dr. Holtzman, a question about sleep. Is sleep important for Alzheimer's disease? Does it have any effect on this amyloid protein that you talked about?
Well, we certainly have evidence in animals that it absolutely is important for the getting good sleep, for preventing the buildup of that protein. And Brendan Lucy, who's in the audience, has also studied this in humans, and it looks like that's true, at least in over short periods of time. Poor sleep also leads to higher production of amyloid. So we think that's a critical factor that you can regulate to try to decrease your risk to get Alzheimer's disease.
Thank you. Dr. Summers, if you're needing participants for this study that you and Dr. Peel are doing, do you want to enroll only people who already have had an audiologic evaluation and are using hearing aids?
We would certainly like to have a broad range of individuals. So just because you've already had an audiological evaluation or you're hearing tested, one of the things that we know is that hearing changes over the course of relatively short amount of time, between even 1 and 2 years. There can be significant changes. So yes, we'd love to have you enroll in a study and give you an updated evaluation of your hearing and your speech perception abilities.
Okay, Dr. Long, you talked about news media reports of infection. Where can people find these reports?
It's as simple as just Googling Alzheimer's and infection. You're going to find lots of news reports. If you're interested in the actual studies, most of them are referenced in those news reports, and you can access them there. But there have been multiple studies in CNN, NPR, Newsweek, as I mentioned. So just search the web; they're available.
Okay, Dr. Buckles, is Alzheimer's disease more likely to affect women than men?
The prevalence is slightly higher in women, but women survive longer.
So what's that mean, women survive longer?
So we're slightly more represented in those with Alzheimer's than males are.
I think what you're saying is because the greatest risk factor is for Alzheimer's getting older, and women tend to get older more than men.
That's right.
I thought that's what you were saying.
You got that right.
Dr. Stark, what about diet and exercise? Is that important in any way for Alzheimer's disease?
Some of the more promising interventions are actually diet and exercise, but that works for pretty much everything. Heart disease, Alzheimer's disease, that's our best guess right now. So exercise and diet are always a good strategy to stay healthy.
Okay, I guess I'll take this one. So we're talking about new ways, online, smartphone, diagnosis of cognitive impairment and Alzheimer's disease. What about the eye? Can the retina, the back part of the eye, show indications of Alzheimer's disease? And again, it's a possibility. One of our participants who has presented here in the past, Dr. Greg Van Stavern from our Department of Ophthalmology, has been studying that. Some of you may have participated in that. And I just got contact from him yesterday. He wants to extend those studies. So maybe next year we'll be able to talk a little bit more about that. Dr. Holtzman again. So the Memory and Aging Project asks its participants to get lots of MRI scans. Is that to track the development of amyloid in the brain? Is that what the purpose of that?
Yeah, MRI scans don't detect amyloid or other proteins building up in the brain. They are used to look at the structure of the brain for the most part. And also, depending on how you do them, you can also look at how the brain is functioning, but they don't measure amyloid.
Okay. Question comes up, and I apologize. Usually I try at the outset to describe this, but a lot of people don't fully appreciate how Alzheimer's disease and dementia relate to one another. Are they different? Is it the same thing? So in a nutshell, dementia means that an individual, a person, is losing their memory and thinking abilities that they had all their life. So all of us have different memory and thinking abilities. We're all different. But for that person, their memory and thinking is deteriorating. And it's not simply a complaint. My memory is terrible today. It's got to be deteriorating to a degree that it starts to prevent that person from carrying out their usual activities. They can't do it as well. They can't drive an automobile as well. They can't do household finance as well. They can't shop as well. They start forgetting. Or they can't reason as well. And eventually, as dementia gets worse, it's not only they're not doing things as well, but they have to relinquish them and other people have to take over for them. Until the end, as the dementia continues to get worse, they can't even do simple things like dressing or bathing or toileting. They total care. So dementia robs a person of their memory and thinking abilities. Okay? Got dementia? Now the question is, what causes dementia? And there are lots of causes. I looked this up once. Over 100 different reports of dementia. Dr. Long mentioned a couple infections that cause dementia. Tuberculosis, syphilis. Lots of causes of dementia. Anything that affects the memory and thinking areas of the brain. Strokes can cause dementia. But the number one cause is Alzheimer's. By far. So 70-80% of dementia is caused by Alzheimer's disease. So Alzheimer's disease, this loss, remember we talked earlier, loss of brain cells, that's the leading cause of dementia, and that's related very much to aging. We have to figure out the connection there. So now, are you with me? So everybody with Alzheimer's, if they live, will get dementia because it destroys brain cells. Not everybody with dementia has Alzheimer's. Does that make sense? Okay. Does that answer that? Because I can't think of anything else to say about it. All right. Dr. Holtzman, are there disadvantages to these antibody studies? Is there a risk? Do they cause problems?
Almost every treatment potentially has risks. In terms of antibodies, some of the antibodies that were generated to amyloid beta have shown some side effects. Some have not. So generally, the companies, nor would we do the trials if we thought the risks were greater than participating in the trials. So yes, though there are some risks. The antibodies to tau that are in development by several companies have not to date shown problems yet in terms of toxicity.
Okay. So a couple questions I'll try to answer. One question comes, if we go to smartphone apps, can I get rid of the spinal tap? No. I shouldn't say that. I mean, by the way, we are so, you, the Memory and Aging Project participants, are the greatest repository of spinal fluid in Alzheimer research in the world. You're remarkable. And I tried to point out that we think the first indication of that preclinical phase of the disease, you know, it's building up for 20 years or more, is detected by the spinal fluid. So if we want to intervene then with drugs, we want to know the first abnormality, and right now it's detected in the spinal fluid. So the spinal fluid is very valuable. But say this smartphone app can also pick that up 20 years, 25 years before, yeah, then we'll get rid of it. So let's check back in 25 years about that. So we talked about our African American Advisory Board, and a question comes up, why don't we also enroll Hispanics, Native Americans, Asian Americans? If we looked at the greater metropolitan St. Louis catchment area, about 13% of all people 65 and older are African American. In the entire population, less than 3% are Hispanic, less than 3% are Asian American. So we can't be all things to all people, so we focus on the greatest underrepresented group in the St. Louis area, African Americans. But as I mentioned, what we find is relevant for other underrepresented groups. If we only study whites, we're only going to learn about Alzheimer's in whites. So we're promoting the investigation of Hispanics, Native Americans, African Americans in research. It's just that we don't have the population to support that, if that makes sense. Okay, Dr. Cummings-Vaughn, your report on the predictive power of an individual who we are assessing in the Memory and Aging Project is fair, but the prediction of that study partner is even better. But people want to know, how do I know if I'm a participant or a study partner?
How do you know? Well, that's an interesting question. I really don't know how to answer that.
I think I can clear it up. If you're asked to have a spinal tap, you are a participant.
That's right. That's right. You're asked different questions, but yeah, the spinal tap, most definitely, there are differences.
The person that we are studying, sure there are questions for both the study partner and the participant, but the person who then goes on to get the memory and thinking testing, the MRI, the PET scan, the blood test, that's the participant. The study partner is usually the spouse or other relative or friend. And we don't test the study partner with all that. Does that make sense? Okay, Dr. Long, do you think if a person does have a bacteria or some viral infection, are they making more amyloid in the brain?
I don't think we have enough data in humans to answer that question. In animal models, there's some suggestion that with infection with various microbes that there might be increased amyloid beta. But as I mentioned, animal models don't perfectly replicate what happens in the human brain. And so we need further studies to answer that question.
Okay, Dr. Zox, what is this memory loss that many older adults experience that doesn't really represent Alzheimer's dementia, but is very pervasive and very frustrating. What is that?
So one of the things I'm struck by is that what does not bring people in to talk to my colleague Dr. Morris is problems remembering paired associate words.
What does that mean?
So, remembering lists of words and lists of pictures of the sort that we often give in the lab, that's not what raises people's concerns about memory. What raises people's concerns about memory is if they're having trouble following a TV program because they can't reinstate the context from the previous episodes, if they're having trouble tracking conversations with friends and family. And so we focus on materials that try to replicate those situations and show how memory is actually used. And memory tasks in the lab often involve a deliberate search process where you're thinking about what happened on a particular previous occasion, and you're thinking about relatively arbitrary materials. Most of the time, what memory is doing for us is not that. I'm not saying that's not important. Sometimes, somebody asks us a question about what we had for breakfast last week, and we have to answer it, and we think that way. But most of the time what happens is I'm in a situation. It's related to other situations that I've been in before. And I use those previous experiences to do better in this new situation. And those are the aspects of memory that we focus on the lab. Both of these kinds, the very unnatural kind and this kind that's kind of automatic and ongoing, are affected by both healthy aging and the early stages of Alzheimer's disease. So understanding the mechanisms by which they really guide our comprehension and ability to perform in real life is obviously important.
Okay, Dr. Holtzman, so very interesting work on tau and tau antibodies. Let's come back to amyloid. Are there, because you mentioned tauopathies can be found in illnesses, you mentioned progressive supranuclear palsy, in illnesses that are not Alzheimer's disease. What about amyloid plaques? Are there illnesses that feature amyloid plaques, or is that specific to Alzheimer's?
There's one pretty common condition called cerebral amyloid angiopathy, and that's where the same protein that forms the amyloid plaques builds up in the blood vessels in the brain. And that will often manifest as somebody having a brain hemorrhage or a stroke. And that's exactly the same protein. Sometimes that happens in people with plaques as well, but it can happen independently. So that's pretty common.
But amyloid in the blood vessels, the angiopathy, do those people have amyloid plaques?
Some do and some don't.
Do they have Alzheimer's? Does that mean they have Alzheimer's?
Not necessarily. Sometimes they can just have that without having Alzheimer's.
Okay. Let me try another way here.
I mean, 90% of people with Alzheimer's disease also have this amyloid angiopathy, but it's usually not very severe in people who develop Alzheimer's disease.
Yeah. What I was going to aim for is that if you have amyloid plaques in the brain, whether or not you have it also in the blood vessels, that confers more specificity for Alzheimer's disease than, say, the tauopathy in the tails.
Yeah, that's true. Yeah.
Two questions about currently approved drugs. Donepezil used to be called Aricept, and memantine used to be called Namenda. And one comment was, do these drugs work? So I would say that if you look at lots and lots and lots of studies, let's talk about Donepezil, also known as Aricept. People who take Donepezil and have Alzheimer's disease dementia don't decline in their memory and thinking quite as fast as the people who are on the placebo, the dummy pill. But that degree of improvement is very, very small. And I think, and again, Dr. Long, Dr. Holtzman can comment on this, we used donepezil because we have few alternatives, but it's really, we're waiting for truly more beneficial drugs. Okay, so I think that's donepezil. Memantine, or Namenda, has also been shown to benefit people with Alzheimer's dementia by slowing their rate of decline compared to people on placebo. Again, a very small degree of benefit, but only for people in the advanced stage of the disease. Now, the company that produces this really tried like heck to show that it benefited people in the mild to moderate stage, because that's where most people with Alzheimer's disease are, and they couldn't ever detect a benefit. So it's approved only for the treatment of people with moderate to advanced Alzheimer's disease. And again, the benefit is very small. Now, lots of people hear about it and start taking memantine or Namenda, often with donepezil or Aricept, in the mild stage of the disease, and I think that helps their urine become more expensive, but it doesn't benefit them. Listen, you are terrific, both participants and study partners. And what a terrific group of panelists. Let's thank them. I'm sorry we couldn't get through all your great questions. We'll try to collate them and see if we can come up with some answers to some of them in the future. There are people and tables and brochures about all of the studies that you heard mentioned, plus more. So please make yourself available. Thank you. Have a wonderful rest of Saturday. Fingers crossed for the Blues.